growth is a general term for abnormal cell growth that can potentially spread to different parts of the body. Cancer occurs along with 6 characteristics that are identified as self-sufficiency in growth signaling, dulling of anti-growth signals, evasion of apoptosis, limitless replicative potential, induction and sustainment of angiogenesis, and metastasis. Most commonly, cancer is simply identified as out-of-control cell growth. The kind of damage it causes include growing and interfering with body functions. Cancer is hard to cure because it evades our immune system by stimulating the protein receptors of normal cells. That is why research is being done today on antibody-drug conjugates, or ADC, to treat cancerous cells. Antibody-drug conjugates are custom manufactured to combine 3 components to target cancer: an antibody, a cytotoxin, and a linker that combines them. First, the antibodies target and bind to specific proteins on cancer cell surfaces that the immune system normally would not. After binding with a cancerous cell and infiltrating via endocytosis, ADCs release chemotherapeutic cytotoxins on the cell. This is unique to traditional chemotherapy, which only releases cytotoxins over an infected area and harming also healthy cells. ADCs target and kill cancer cells discriminately. ADCs can be thought of as a biological homing missile that is guided to its target in order to deliver a deadly payload.
Antibodies are immune system proteins that are responsible for finding and tagging antigens. These proteins each consist of 4 polypeptides. These 4 polypeptides come together to form a quaternary Y-shaped structure. The tips of these Y-shapes are where the antigen binding sites are. Thus, the tips have the greatest amount of amino acid sequence variability so that they can be made to adapt to all different kinds of antigens. This region typically consists of 110 to 130 amino acids, giving the antibody the necessary binding specificity. Tips are known as the variable regions, which are subdivided into hypervariable HV regions and framework FR regions. The HV regions directly contact antigens, while the FR regions form a beta sheet, which acts as a platform in order to hold the HVs in position to contact antigens. There are many possible ADC linker candidates that are currently being researched, each one designed to be cleaved or to release the attached toxin in different ways. These candidates include hydrazone and peptide linkers. Hydrazones are organic compounds that consist of an amine group, a nitrogen atom, a carbon center, and 2 side chains. Due to their high sensitivity to low pHs, hydrazone linkers are designed to degrade within acidic cellular compartments. Peptides are chains of amino acids which consist of a central carbon atom, an amine group, a carboxyl group, a hydrogen atom, and a side chain. Peptides are commonly occurring biological molecules that can be degraded by cellular lysosomes. Thus, peptide linkers are designed to be enzymatically hydrolyzed by lysosomal protease. Hydrolysis being a reaction that breaks a bond in a molecule using water. The enzymes within the lysosomes act as catalysts that speed up the reaction but are not consumed by it.
Cancer treatment is one of the most needed solutions in society today. However, in order to rid of cancer cells, strong and powerful chemicals such as monomethyl aristatin E are required to terminate these substances. The only problem with these types of drugs is that they are too powerful and can ultimately damage not only tumor cells, but as well as healthy cells. Therefore, with the aid of attaching a specific drug to the antibody, it reduces the risk factor of terminating healthy cells altogether. Although the ADC drug is said to be activated only at the tumor site, one problem that engineers and researchers have faced is the fact that the first linker can break, which then releases the drug from the antibody prematurely. This can then result in the death of a healthy cell and ultimately lead to peripheral neuropathy. Engineers, in order to accommodate this problem, are in the process of creating more stable linkers so that the linkages don't break at the utmost critical moment. Another issue that researchers have found is that while the drug travels to its specific target, healthy cells can ingest the drug conjugate. Fortunately, for some, the cell is able to recycle these unwanted chemicals. But on the downside, if the cell ingests too much and is unable to recycle this substance, it then leads to the death of the cell, namely apoptosis. Thus, researchers are undergoing trials where they develop ways to make sure that the drugs only target the right cells. In recent trials, nearly more than 24 ADCs are being tested in order to find the right combination of the drug, antibody, and linker to ensure that it hits only the tumor. Lastly, another problem that researchers are facing is the fact that although conjugates reach their specific target, that being the tumor, it doesn't necessarily mean that it will terminate the cell. The goal of an ADC, however, is that it will eventually eliminate the tumor cell. But if it fails to perform its given task, all that results is the dumping of chemicals into the body, which can have a major effect on the duties and efficiencies of other cells. Efforts to fix this problem is ensuring that the protein that is selected as a new conjugate target gets internalized into the cell. Thus, the future and goal of ADC is that it will bind to its specific target without harming other healthy cells and combine to the cell surface, which will generate a complex that will be internalized within a cancer cell in order to spare the toxicity of the patient.